there's a danger lurking here on the ground. It's left its mark in the fields, too. And it could even be hiding here, a threat to our lives among the innocent flowers? Flower bulbs like these are apparently dangerous. Disease-causing fungi have supposedly developed from them and killed people. Invasive fungi can enter our bodies and, in the worst cases, kill us. They are also spreading worldwide. The threat they pose is growing significantly because our means of combating them are less and less effective. This applies to agriculture too. So, what's in store for us? Here at Cologne University Hospital, I learn what it means to contract an invasive fungal infection. Dogan Eskachurek has been infected with invasive fungi, which are growing in his head. For months, he's been receiving infusions to stop the fungal growth. The strong medication's side effects are also taking their toll on him, straining his liver and kidneys and making him weak. Dogan whispers something.
Wir kämpfen zusammen.
We're fighting this together.
Wir kämpfen seit Monaten zusammen.
For 9 months, I have only been concerned with him and with his illness.
At first, the infection wasn't recognized, and then it was underestimated. In the meantime, the fungus has spread from his sinuses further into his head. Dogan describes how it feels like to have a fungus in your head.
Ağırlık var. Ağrı var, kaşıntı var.
Very severe headaches, and it itches everywhere, around the eyes, the nose.
Zevki Eskachurek searched everywhere for a specialist for her husband. Another clinic was unable to help him, and the fungus continued to spread. Now he's here in Cologne, where doctors are pushing his body's limits with medications in order to save him. And it finally seems to be working.
After 3 weeks, they told us today that the blood analysis results are now okay. So at least we're on the right track.
But fungal infections are extremely persistent. In the field of invasive fungal infections, Oliver Corneli is one of the experts in Germany. Every day, he's confronted with the most diverse cases. He shows me how difficult treating fungal infections can be. The X-ray shows how the fungus has already grown from the sinus deep into the head.
And the brain is on the other side of the sinus wall. If the fungus grows through the bone, it goes directly into the brain.
So the fungus is already very near the brain?
Yes, it can break through into the brain at any time. We cannot let that happen. Which is why we've prepared to take such risks with this treatment. It's better to risk damaging the kidneys or the liver than the brain. The kidneys and liver can recover, but the brain cannot recover if the fungus grows into it.
Dogan Eskachurek is not an isolated case. Fungal infections and their treatment severely restrict patients' lives. They live with daily infusions, pain, fatigue, shortness of breath, and organ damage.
Sie atmen ganz normal weiter erstmal.
Those affected often present very late. Too late because the fungal infection had not been detected. People whose immune systems are already weakened by other diseases are most affected. the door is essentially open to the fungus. People with poorly controlled diabetes, tumors, transplants, or who are receiving cortisone treatments are also at great risk. Fungi always exploit immune system weaknesses to spread. Even mold can become life-threatening. This is because, as shown here, it can also grow in our bodies. Fungi can be found virtually anywhere where organic material decomposes, like in compost or in the forest. The fine heads at the hair ends are filled to bursting with spores. When they break open, thousands of spores are released into the air, which can enter our bodies in various ways. Through open wounds, into the bloodstream. If inhaled, they can settle in the sinuses and grow into the tissue. Very often, they reach our lungs. If the immune system does not slow them down, asthma-like symptoms or pneumonia can result. If their growth isn't stopped, the fungus finally reaches the blood vessels via the fine air sacs in the lungs. It can then spread quickly throughout the body. If it binds to a vessel wall, the fungus grows into the organs, clogging the blood vessels and eventually overwhelming them completely. The fatal result is that wherever blood flow is interrupted, medication can no longer reach the affected area. Every year, at least 2.5 million people die from fungal infections. But what exactly are fungi? They're neither animals nor plants, and yet they're everywhere, and there are at least 2 million different species. Although some threaten our health, life without them would be impossible. Fungi break down all dead matter into its basic elements so that new life can keep emerging. Otherwise, piles of dead wood and old leaves just remain. Breaking down matter is one of the fungi's most important jobs, but in our bodies, it's especially dangerous. Specialists used to believe that fungi were only harmful to those with weakened immune systems. But fungi proved not to follow our understanding. Every year, thousands of people journey to Vancouver Island, off Canada's west coast, to marvel at the natural beauty. But in the early 2000s, a series of puzzling illnesses broke out in this idyllic setting. At first, dogs, cats, and wild animals were affected. Then, unexplained illnesses were reported more and more frequently in humans.
People were getting sick from this. Were getting very sick from it, and some people were dying. So we definitely wanted to find out as soon as we could what the source was, why people were getting infected, why some people were getting infected and others not.
Dr. Murray Fife was part of a team of physicians and researchers who set out to find the cause.
So we were finding actually most of the infections were in people who had competent immune systems, that didn't have weakened immune systems through disease or medications. And that was a concern, right? Because we didn't expect to find that here.
Ken James, for example, loves spending time outdoors. Then, while not yet 50 and in very good shape, he began to experience strange symptoms.
A constant headache, just day and night, night sweats. I sometimes had to change the bedding twice a night because we're just sweating like crazy. The fatigue was a major thing. I had problems in my throat, kind of like a constant dry cough.
His doctor examined him, and the X-rays showed 2 unclear shadows on his lungs. Ken was terrified that it could be cancer. But things turned out quite differently. Tissue samples revealed the cause of the strange X-ray findings and symptoms. It was a Cryptococcus gattii infection. Normally, this fungus does not pose problems for healthy people.
But some people develop a lung infection, and from the lung it can also spread to other tissues, including the brain or the lining around the brain, the meninges.
But how were all those people infected with Cryptococcus gattii? What was the source? Murray Fife and his research team took samples of soil, dead wood, bird droppings, and air from all over the island. They finally found the fungus on the mighty Douglas fir trees on the island's east coast. From there, it had dispersed its spores into the air. Everyone nearby had inhaled them.
So it could be people who work indoors or outdoors, people who do a lot of gardening or don't do a lot of gardening. Basically anybody would be susceptible to this infection. It's not limited to only people who are immunocompromised.
Thousands and thousands of people had been there that same weekend, and I'm the only one that came away with it, so, you know.
After being correctly diagnosed, Ken was given the right medication and was back to full health in a few months. But between 1999 and 2007, a total of 19 people in British Columbia died from the fungus. More than 200 fell ill. Vancouver Island demonstrates how dangerous fungal infections can be, but also that it can be effectively managed with medication if detected early enough. Back in Cologne, I wonder how successful Europe is in treating invasive fungi with medication. Dogan Iskichirak is living proof of why medical treatment should be started early on. Otherwise, the fungi become deeply embedded in the tissue, and treatment can take months or even years. But infectious disease specialists like Oliver Carnelli are struggling with antifungal medications. Fungi mutate quickly and become immune to the drugs.
They've already developed some resistance. We have to be careful that the fungi don't become uncontrollable, as has already happened with bacteria.
Oliver Carnelli wants to explain the problem to me in more detail in the hospital's microbiology lab. Here, fungal pathogens from infected patients are examined to determine which drugs counteract them. The fungal spores are cultivated in petri dishes. The test strip carries a drug. The small white fungal colonies keep their distance because the antifungal drug is effective against them. If the fungi grow on the strip, like in this case, they're already resistant to the drug. That means the drug is no longer effective.
Every day we see patients with exactly the same problem. Every day we contend with the issue of resistance.
Anna Dudakova, the lab head, shows us a patient's test results. Several antifungal medications were tested. The standard medication is shown in the upper right corner. The fungus has completely overgrown it. It's no longer effective.
This is our standard medication, which would mean complete treatment failure in this case, as this fungus is immune to this drug.
But in principle, these two should work.
Yes, they should work.
If we didn't have this lab result now, we would continue with this standard treatment and would not understand why our patient's condition continues to deteriorate, and she ultimately dies.
Fungi are masters of adaptation, which makes combating them so difficult. They're already resistant to many medications. But how? Why are invasive fungi becoming more immune? In search of an answer, I'm at the University of Wageningen in the Netherlands. Researchers here have discovered something shocking. A few years ago, geneticist Evelyn Snelders and her colleague Simon Schuster were investigating a series of life-threatening fungal infections. At the hospital in nearby Nijmegen, the drugs failed to work against a specific type of mold. The mold was resistant to them. Normally, such resistance only develops when drugs are used frequently and over a long period.
If you treat patients for long periods of time with an antibiotic or with an antifungal, resistance can be selected for. But this was already different at the start.
None of the patients at the hospital had ever been given an antifungal before, so the resistance could not have been induced by medication.
So I think we already at the start knew there must be a different explanation.
Evelyn first had to rule out the hospital as the possible source of the resistant fungus. If it were the source, all the patients would have been infected with the same fungal variant. But her genetic tests revealed that the patients were infected with different variants of the dangerous mold.
So then it became clear that it was not one source from our own hospital, but the source had to come from elsewhere.
But where did the resistant fungus spores come from? That was when environmental geneticist Simon Schuster came into play.
And then we thought, okay, what is the natural habitat for this particular fungus? So that is dead plant material. So all plants that are decaying, are dying, are dead, they all get sort of decomposed. So this fungus is one of the major fungi decomposing those materials.
The team conducted a thorough search. At that point, they believed they were looking for the proverbial needle in a haystack. How could a fungus outside the clinic have become resistant to drugs? All conceivable sources of mold were checked: grass clippings, flower beds, compost heaps, fields, greenhouses, garden centers.
But it was quite shocking that it was so easy to find them in the environment too, because we were first very focused on the clinical isolates, not realizing if you would just go outside, you could also retrieve a resistant isolate.
DNA analysis of the samples confirmed that the source of the resistant fungal infections must have been outside the clinic. But for environmental fungi to have developed resistance to drugs, they must have come into contact with the agents. And then they finally found the crucial clue. Agricultural pesticides were the source.
The drugs that are used for patients are chemically the same as the fungicides that are used in the environment. And that's special.
Stop. That's all the same substance used to treat us.
The drugs that are used for patients are chemically the same as the fungicides that are used in the environment. And that's special.
And at the time that was actually new for us. They have slightly a different name, but they are the same chemical group.
The explanation: contact with the pesticides allowed the fungi to become resistant to the life-saving drugs. When plant waste from sprayed strawberries, potatoes, or flower bulbs is dumped onto compost piles after harvest, the fungicide ends up there too. The molds on the compost come into contact with the same substance used to treat hospital patients. That means only those fungi that happen to be resistant to the substance survive exposure to the fungicide. These then multiply unhindered and release their resistant spores into the air, which everyone breathes. The most important drugs are now ineffective in patients infected with these spores. That's why, at the time, help came too late for some patients at the Dutch hospital. This finding was so inconvenient that at first, no one wanted to believe the researchers. But eventually, samples throughout the Netherlands were examined. All of them confirmed the results.
So at first it was really good that we got recognition. And then of course you also realize that it's just the tip of the iceberg.
The results show that while the resistant fungi spores are in the air everywhere, there are hotspots. At facilities where large amounts of green waste from intensively sprayed fields are composted, the spore concentration is 20 times higher. These are mainly regions with many greenhouses and strawberry and potato fields. Flower bulbs also play a role because they're treated with fungicides before a sale and end up in the compost along with the pesticides. Flower bulbs that have been treated with fungicides indeed play a role. What I find so striking is that the real problem is much bigger. Given the dramatic consequences, why are these pesticides not simply banned from agriculture or at least used more sparingly? During my research, it quickly becomes clear that fungal infestation is also a huge problem in agriculture. Fungi threaten entire harvests. I'm meeting with botanist Eva Stukenbrock in Kiel, who has declared a war on fungi. She first takes me to Hohenschulen, an experimental farm belonging to the University of Kiel. Stukenbrock wants me to see these fields because they are the cutting edge of pesticides in use. I'm curious to see what awaits me. Farmer Jill Jensen brings us to a barley field. The barley has finished growing and will continue to ripen until harvest time. Jill picks an ear from the middle of the field. These plants received the full dose of fungicide when they were sprayed.
Danke, Jill. Ja, so guck mal. Ja, das ist jetzt vom Mitte.
This one is from the middle of the field, where they were properly treated.
Richtig behandelt wurden.
We compare these plants with those from a corner of the field. Pesticides are usually not applied there because these areas are hard to reach with the large sprayers.
There's more fungal infestation. Here you can see with and without fungicide. You can clearly see the difference between the two barley plants.
The ear infested by the fungus is considerably smaller. Fungicides seem then to be indispensable in agriculture. Then, Eva takes me to the fields outside the research farm. Here, too, fungicides have been sprayed, but they're no longer effective. If you look closely, you can see yellow and brown spots all over the leaves. This is where the fungus has penetrated and the leaf cells have died. The leaves can no longer absorb the sun's energy here, which they need for growth. As a result, the ears are much smaller. Every year, global farming loses at least 20% of its harvests to fungi because the pesticides are no longer effective. Back at the Hohenschulen experimental farm where Jill works, I want to see how they control the fungus problem.
This is the induction hopper, which has just been lowered.
So this one is in use and you can switch to this one? The key is to combat the fungus with a range of different substances that are precisely formulated to work together.
I'm setting it up the way we used it this spring.
The idea is that if fungi are resistant to one fungicide, another from the blend will destroy them. But the experts think this elaborate process isn't really used much in other parts of agriculture. I look at the substance names more closely. Many end with azole. Here's another azole.
That's right.
Later, I'll look into whether these are the same substances used in medications. Eva is convinced that we cannot solve the fungus problem with fungicides alone. She wants to show me why the fungus grows so easily in our fields. That's all wheat?
This is all wheat. It's a monoculture, meaning only one variety is grown here. All these plants are genetically identical, clones. That means that if the fungus infects one plant, it can infect all the plants in this field.
At the university research facility, Eva wants to show me how fungi can be kept in check using fewer fungicides. This simulates the situation in the field, a monoculture where all the plants are identical. The fungus only needs to overcome the immune defense of one plant species in order to infect the entire field. When different barley types are grown in the same field, it's not as easy. To infect the whole field, the fungi have to overcome the defenses of all the different species. They can rarely do this, and the crop is much better protected. The variety includes especially resistant species, which act as barriers and further prevent the fungus from spreading rapidly. In Denmark, such mixed crops were already being used for more than half of the wheat growth in 2024, with no loss of yield. The process is still in its early stages. Eva says it can be improved, but it clearly works. The idea is that this will reduce the use of fungicides. Does it work?
It works. First of all, there is 30 to 40% less disease in the wheat fields, which means there is less need to treat them with fungicides.
So it's worth it.
If you can reduce spraying by once or twice per year, you save a lot of money.
We urgently need such solutions because our chemical arsenal to combat the fungi is running out. Back in Cologne, I want to know more about antifungal agents and medications. How many substances are there? Andrea Liegweg, head of the hospital pharmacy, collects all the available medication for us. Lots of drugs, but no antifungals.
Oh yes, there are a number of them. Here's micafungin. Let's sort them.
The box is half full now.
Azole, 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 azole.
We mainly find azoles. This time not in canisters, as I photographed them in Kiel, but in medicines.
Here you can see the suffix, azole. Azoles refer to the substance group. Azoles are used very intensively in agriculture, and they are also the largest substance group in treatment for humans.
But what is used here in milligram or milliliter quantities is used in agriculture by the kilogram and liter, so resistance develops much more quickly.
If, for example, the fungi develop resistance to azoles, as we are increasingly seeing with Aspergillus, then none of these drugs are effective anymore. That means there are now only 2 options left. Most of these have not yet been approved and have not been properly evaluated for Aspergillus, to stick with this example. So very quickly, from the original 3 options, only this one is left.
If these medications become ineffective too, will we then be completely defenseless against the fungi? Near Trier, I learn what dramatic consequences this could have.
Let's turn our lamps on.
Researchers Stefan Lötters and Amadeus Plevnia are tracking a dangerous invasive fungus. Fortunately, it doesn't kill humans, but it has a devastating impact on native amphibians.
Chytric fungus, or bezel, literally eats holes in the skin of salamanders and newts. You can see that this salamander has little holes everywhere. This one is also very badly affected. That must be very, very painful. Here it is again.
Those are real craters.
Exactly. And they open the door to bacteria. The animals often die from secondary infections, not from the fungus. The fungus suppresses the salamander's immune system.
Does that mean the fungus is transmitted from animal to animal?
Exactly. Like during mating season.
It is very rare and dangerous that a fungus is transmitted through skin contact, which accelerates the spread. But why is the fungus suddenly such a big threat to our salamanders?
This fungus has been introduced from Asia, where amphibians are resistant to it. They have evolved alongside this fungus over millions of years. Here, the fungus is new and invasive. It doesn't belong here. The amphibians have no immune response to it.
Lötters tells me that around 100 species, mainly in South America and Australia, have already died out due to invasive fungi. A fungus with similar characteristics to the one threatening fire salamanders has also become acutely dangerous to humans. It appeared seemingly out of nowhere and is also transmitted through contact. Called Candida auris, it is spreading rapidly and is resistant to most medications. Candida auris was first discovered in Japan in 2009. It soon spread to several continents. And it's reaching ever more countries. Alongside India, South Africa, and Venezuela, the US has been very hard hit, with more than 4,000 infections reported, and the numbers are rising fast. Candida auris is difficult to stop. And it's reached Europe. Several hospitals in the Valencia region have experienced major Candida auris outbreaks. One of the affected hospitals is here, very near the coast. The La Fay University Hospital is the region's central medical provider and an important center for organ transplants. Many seriously ill people with compromised immune systems are treated here. When the first unexplained infections appeared in the intensive care unit in spring of 2016, the team that later became involved in the Candida auris task force had no idea what was in store for them. The ward had many patients with weak immune systems, so the appearance of fungal infections was not surprising at first, but that would change. The patients were given the usual antifungal medications, but they had no effect. The fungus seemed immune to them.
So we were scared, really scared, when we realized what was going with the patients, because they remained positive in the surveillance cultures, but also they become infected, some of them. And when they become infected, this was really awful because they were really severe.
Disinfectants were also ineffective at stopping the spread. The laboratory microbiologists were baffled. Had they misidentified the fungal pathogen?
The problem with this new species was the same worldwide. The new species had not yet been recorded in the pathogen identification databases. As a result, it was misidentified, and everyone reacted incorrectly.
Only when Dr. Payman and his colleagues ran a genetic test did they realize that they were dealing with Candida auris, a new fungus about which little was known. That is why the drugs weren't working. Concern was running high at La Fay. The fungus continued to spread in the intensive care units. 3 of the first 4 patients died, and shortly after, 22 more were infected.
The aim was to control the spread of the outbreak and save as many patients as possible. We also tried out new treatments and introduced new disinfection measures.
The affected patients were isolated and monitored continuously. All hospital room surfaces were disinfected twice a day, but to no avail. Candida auris continued to spread, and the source remained elusive. The task force of microbiologists, infectious disease specialists, and intensive care physicians ordered that swab samples be taken from every possible contact surface. From walls, floors, tables, medical equipment, and from staff, patients who were not yet infected, and in operating rooms. The laboratories evaluated hundreds of samples and finally obtained results.
We didn't find Candida auris on the employees, but on the walls, tables, beds, and blood pressure monitors.
They also like the catheters. So if we use catheters, intravenous lines, and all these things for the patients, they were attached to the plastic. So we have to remove all the catheters, but also there were other devices that were invasive, that were not easy to remove.
More than half of those who were infected died. Many specialists, including from abroad, were now searching for life-saving methods and substances. In the end, a cocktail of medications helped those infected, and chlorine-based agents were used for disinfection. The hygiene routine for hundreds of staff had to be completely changed. It took 2 years for the measures to finally take effect and the number of new infections to decline. The fungus has not completely disappeared. It continues to hide in the smallest corners and in or on patients. But now there is a way to deal with it.
We didn't know how dangerous it was, so now we know it. And we hope that if this affects all the hospitals, all the countries, they will learn about our experience and be really quick with the first with the first patient.
La Fay remained on high alert, with patients and surfaces constantly screened for fungi to prevent Candida auris from spreading. But it would remain in the clinic. I wonder if other hospitals have learned from the experiences here in Valencia. After all, the fungus can turn up anywhere. Pathogens can now spread very easily over long distances. There have already been several minor outbreaks in Germany. Only the National Reference Center for Invasive Fungal Infections can tell me if we're prepared for larger outbreaks. All Candida auris cases have to be reported here. Oliver Kurtzai and his team are the experts for all difficult questions concerning invasive fungi. They're the point of contact for all hospitals and laboratories in Germany. If anyone has an overview of the state of risk in the country, they do.
In 2023, there were around 80 cases of Candida auris in Germany. That's why we made a concerted effort to contact diagnostic laboratories, which is also part of our job as a national reference center. They have to step in and tell the hospital when there's a new and dangerous pathogen that requires action.
80 cases doesn't sound like many, but that's only the number of people who are reported ill. Kotsai says no one knows the real figure because there's no general reporting requirement. And climate change could worsen the situation because the fungi can adapt to new conditions.
This adaptation to higher temperatures could lead to fungi infecting humans more often. This is one of the most popular hypotheses as to why Candida auris is suddenly on the rise. If it's true, then we should expect many more fungi to follow suit.
So we should urgently prepare ourselves for new invasive fungi, because once they've spread, they're very difficult to combat. All examples show that a rapid and comprehensive response is required, whether in the body, in a hospital, or in the fields. The antidotes we have are already inadequate. Now it's important that new drug substances are developed. But we cannot use these substances in huge quantities in the fields, where fungi quickly develop resistance to them. We have to realize that invasive fungi exploit every vulnerability we offer them. Like agricultural monocultures, which literally invite fungi. More intelligent farming methods could offer a solution while also reducing the use of pesticides. Invasive fungi pose a real threat. If we want to avert it, we have to seize every opportunity.